Hello and welcome to PiFM at Work. This video is part of a collection of videos where we highlight cutting-edge applications using the VistaIR microscope from Molecular Vista. Now in this video, we are going to investigate a polylactic acid and an acrylic rubber nanocomposite. And so we're going to be able to use our PiFM technique to deconvolve both two materials, both spatially at the nanometer scale and spectrally at the nanometer scale, showing spectral results that are very much like FTIR results. And so for the next few minutes, we'll start out with an introduction to the sample, followed by a short imaging session just to familiarize with the instrument, followed then again by some data analysis highlighting the spectral and spatial capabilities of the tool, and then we'll wrap it up with a short conclusion. All right, well, with that, let's get started. Let's start out with the importance of polylactic acid. Now, polylactic acid is formed from a lactic acid, and it's usually a product of biomaterials derived from starch fermentation. Now, there's a lot of excitement behind this type of polymer because it provides a biocompatibility that you don't see in traditional petroleum-based polymers. PLA is really primarily sourced as a material for bioplastics or things that are going to decompose well. Now there are some intrinsic disadvantages to PLA by itself, and these primarily are in the form of gas transmission through the material and the mechanical properties. So they tend to be a little bit too soft, the rigidity is not there, and they tend to allow too much gas to flow through the material, which of course if you're using this for food storage or something of the like, of course that's not going to work well. Now we do see PLA used pretty heavily in 3D printing because it has a low thermal melting point, and we also see it heavily used in medical implants because the PLA will break down in the body into a lactic acid, which is intrinsically not toxic to the human body. But the other primarily usage is something you've used probably on a recent basis, and that would be disposable or compostable cups, plates, things like this. Now you could envision scientifically that people would want to try to modify or introduce other materials into the polylactic acid to help improve the mechanical rigidity or maybe some of this gas transmission issues. And so this would be a driving factor of why we might want to look at composites of PLA and other materials. Now, as I highlighted in the beginning, we're going to be looking at a PLA ACM sample, so it's going to be a blended sample. And we know that it has the two main components of the polylactic acid, the PLA, and the acrylic copolymer, the ACM. Now, the unknown is how are they distributed at the micrometer scale and the nanometer scale? And that's something that PiFM will do a very good job in helping us resolve. So our objective in our short imaging session we'll do together is to resolve spatially the two components of the PLA and the ACM and identify them by PiFM spectra, and we can do some FTIR comparisons from literature. Starting out in the VistaScan software package, this is where we'll do our data analysis of the PLA ACM composite. So we start out with our user session here. We have our username, and so that we have MVI demo, sample name PLA ACM, cantilever type, where we're going to save that information, and some detailed information about the imaging modes that we're going to apply here. So effectively, this is where you're going to start and put in your metadata for that imaging session. Now we switch over to the AFM Setup tab. Here we've defined our first and second mechanical resonances of the cantilever so we can do the PiFM modality, and you can see the sample defined underneath the cantilever. So that'll be the imaging area. Okay, then we'll have the cantilever engage with the sample surface. Then we'll be able to go ahead and take a point spectra. So here we'll take a survey spectra just wherever we happen to land. So we'll switch over to spectroscopy. And here what we'll do is we'll go through and take that point spectra, and you'll see that result here below where we have the intensity versus wave number of that area in the sample. Using the representative peaks that have shown up, we can start to make an intelligent guess on where we want to do some of our fixed line imaging. And so we'll do that next. What we'll do is we'll switch over to a wave number that will be associated with the PLA. This will be the 1750 wave number, and we'll start taking a topographic image. So you'll see as the image is coming in, areas that are light or bright are going to be areas of high absorption, or the PLA material. The images that are dark, the areas of the image that are dark, will be that ACM material. And we just happen to know that because we've already looked up some information about the wave numbers associated with PLA and ACM. So we can let this image a little while just to kind of see the image build up, and we'll go ahead and take some images outside of the video session here that we can further analyze. And so now let's take a look at the data set that we acquired on the PLA ACM sample. So to start, let's get a frame of reference. Our top image is the topographic layout of the sample. So we got about a 3 micron scan field, and what we did here is we just did a raster scan with the AFM probe, and then took a point spectras at 3 different locations that topographically look interesting to us. So we noticed that there were these craters, and so inside the craters we took spectra at points 1 and 3, and then in the kind of plateau regions outside of the craters we took a spectrum number 2. So you can look at the spectra here in our spectra analysis window, and we have the 3 locations highlighted. So red being on the plateau, and that has a peak around 1755, which is shifted from the 1730, 1728 peak that you're seeing right next to it here. And so what we know from looking at literature is that the PLA is associated with the 1750 region, and the ACM is associated with the 1730 to 1728 region that we ended up taking images at. And you can also see that there's a lot of spectral differences down in these lower ranges here between about 1,000 and 1,300 wave numbers. So with that information, we could then go back and do PiFM scans where we keep the laser constant. And so what we do is we pick a single fixed wavelength, 1728 for example, for this image, and we highlight the ACM when we raster scan that image. So these images take roughly about 5 minutes apiece. Now you'll notice that these craters all highlight nicely as being locations that are rich in the ACM material. Now if we go and tune the laser to a slightly different wave number, let's say 1750, then we can then highlight that plateau regions or the areas outside of the craters. Now for a little better visualization aid, we can then go and look at those 2 images and combine them visually based on color. So for our ACM locations at 1728, we highlight that as green, and you can see that brought in over here on the combine window. And now what I'll do for the PLA is I'll highlight that as red at 1750, and you can now see how those materials are distributed on that sample surface. Cool thing here you'll notice is that these ACM locations also have these small little divots of material or little pockets of material distributed out throughout the PLA region. Now what was interesting in analyzing this data the first time is we noticed that there is something going on in these black dots. They're not either PLA or ACM specific, and so we decided to go in and zoom in on that region as part of our imaging session. So you can see, let's go back to our spectra, and if you come down to this region here, here's our topographic image that we took in that zoomed in region. Here we're working with about 650 nanometers of a square image, so we're really below a micron at this point. And we did a fixed line image at the 1750. We already knew that that was going to be a good one for the PLA. So we moved down and we then also imaged at 1720 for the ACM. And now we can take some point spectra in those 2 different locations. So here we looked at just the 2. We look at the number 2 label location, which is the ACM, and the number 3 location, which is the PLA. And you can see the spectral differences really nicely highlighted out in that 1,000 to 1,300 region. So then what we decided to do is go and take a point spectra on one of the little black dots. And so that's what you're seeing here at point number 1. So here you see that it's kind of almost like a distributed or splitting between the 2 peaks that we saw for the ACM and the PLA. But what we do notice now when you go into that 1300 to 1100 region, two definitively new peaks associated with that particular material. So is it an unknown? Is it some kind of quasi material that is part ACM, part PLA? That I'll leave to the scientists who are working with the materials. But you can see how PiFM can really get in there, that very small region, highlight it very well with just in these little particles that are only 50 nanometers across or so, and show you spectrally what those materials are or what the differences is from the PLA and the ACM. So really powerful ability for PiFM to do that. So with that, I want to move on. We'll take this material, we'll go and do a short analysis of it in a little PowerPoint slide where we then try to look at the known FTIRs of the PLA and the ACM as a comparison. All right, let's take a quick minute to review the data that we just analyzed in Surfaceworks. So Surfaceworks has a really powerful feature in that it allows us to directly export the images that you were seeing in the Surfaceworks package directly over to other packages of software. So Microsoft Word or Microsoft the PowerPoint programs, no problem, just a quick copy and paste. Same for OpenOffice, we're able to do that same copying and pasting. Now the pasting of those images, they do come in vector-based, so it's very easy to rescale them. And also you can ungroup them and then modify the text if necessary for publication. But these images are publication ready and publication quality. So here I quickly exported the 1750 wave number image for PLA, the 1720 for ACM as in green, and then the blue image, which was our unknown at 1070. I then used Surfaceworks to help me combine those three images. So we have a nice little feature that rapidly lets you combine those, as you saw earlier in the video, and then I overlaid those on the topography image. And that's the 3D representation you're seeing on the left-hand side. So that is the combined image laid on top of the three-dimensional topography image. So then we are able to do the same thing with the spectra. So here is the spectra that we were analyzing before in the video, but now I've quickly exported that into PowerPoint. And so this was our zoomed in image at 1070 for that unknown component, and then the spectra that we took at those three different locations. And so like I said, that all is very nicely brought over. It's all vector-based, so it is very friendly when you want to modify that for publications. Okay, and then in conclusion, what we wanted to look at was how the FTIR of these materials, the known FTIR, compares with the PiFM results that we obtained. And so here you can see that comparison. We have an FTIR for the PLA on the top, and then one for the ACM on the bottom. These were both extracted from literature and known materials. And then what we do is we compare those directly with the PiFM. So you see there's a really nice agreement between the PLA and the ACM's FTIRs and our PiFM result, and it really does allow us to correctly identify that we're seeing both the PLA and the ACM spectrally. Okay, well I hope you enjoyed this short video where we took PLA and ACM nanocomposite and deconvolved it spatially and spectrally using the PiFM technique. I think it really shows the power of the PiFM technique to be able to go into 10 to 20 nanometer areas and get a good FTIR-like spectra that you can use to investigate these unknown materials and known materials and their composites. So with that, I'd like to give a special thanks to Dr. Rudiger Berger. He provided these samples. He's part of the Max Planck Institute in Germany. And also a special thanks to you for your interest in PiFM and watching our videos. I hope you enjoyed the previous videos and future videos to come. But I really want to encourage, if there's something you're seeing in the PiFM where you think could really help your research, to contact us, and we can have a short discussion on how we can apply your application to our technology, and we can work together to really see PiFM pull out scientific results that were untouchable before. So again, I do encourage that. You can reach us at the info@molecularvista.com, and we'd be glad to have that conversation. Okay, well with that, I'd like to thank you again, and we'll see you next time.